Okay. Okay, so this is that tissue activator TPA. This is that gene that was generated by the exon shuffling. Well, we had previously mentioned exon shuffling. This is the example that goes with that. I know, weird place to leave off, but it was what it was. Okay, so this is the portions of the ancestral genes. This is the ones that will be shuffled together, right? And so the epidermal growth factor gene with multiple EGF exons. Here we see an EGF exon. This is going to experience exon shuffling, which will generate the TPA gene as exists today. This is the epidermal growth factor gene. This has multiple finger exons, and this is the finger exon here. And then right here we have the plasminogen gene. This has the Kringle exon, and that's the K, Kringle. And here we see that the exon was shuffled in, and then it apparently was duplicated later on. Okay? And so we get our modern TPA gene. Woo! Interesting, right? Okay, so we also have alternative splicing, of course, which is the other one we had mentioned in that slide previous to the previous one. That's going to be alternative splicing is going to provide the means of forming a set of proteins that are variants of a basic motif without requiring separate gene for each protein, i.e. you have a portion of the gene that codes for that motif, and then you just alternatively splice it into different overall genes. And so right here we have the membrane-bound antibody molecule. This has an extracellular side is over here. This is the cytoplasm side right here. So to bind the membrane-bound antibody molecule, we have to have something that will anchor it into the cell membrane, right? That way it can actually be bound there. This is the membrane anchoring unit. This is actually encoded by a separate exon. This would be the soluble version of that antibody molecule, i.e. the one that is secreted and not actually membrane bound. See, it won't be attached, okay? This is not being cut off of. See, there's no cell membrane bound at all, okay? By the way, does anybody know why this is kind of dipped into here? Remember we were talking about they're secreted from like the ER complex and they have this little cell membrane around them essentially? Yeah, that has joined to the cell membrane here. Yeah, it's those little details that tell you your book is actually really good. It's really, really good. I would never be able to do that figure that nicely. Okay, so this is, again, the membrane-bound one. This is the soluble antibody molecule. And so what we're getting is alternative splicing of our RNA. This will exclude the membrane-bound anchoring domain in the soluble antibody one. In the membrane-bound one, we include it. And so when it tries to be released, it instead binds into the membrane because it has that membrane-soluble portion. Okay? Okay, does anybody have any questions there? Okay, and that's the chapter. All right.